Yes, the title, Small is Beautiful and Why Cell Size Matters. And I will talk about these two issues both on an ecological and evolutionary timescale. But let me just start with jumping in the small is beautiful aspect. Here we see a scanning electron microscope image of maybe the most well-known and poster child of all coccolithophores, Emiliania huxleyi. You see a single cell that is actually hidden behind multiple layers of coccoliths, and these coccoliths are these oval structures that are imbricated around the cell, and these are made out of calcite. And so the coccoliths in this case are about 3 to 4 micrometers in diameter, and the cell that resides within this coccosphere is about 5 to 6 micrometers. So small is beautiful. Why would we actually be interested in such tiny creatures? Well, the good news is that when we zoom out, and in this case look at the satellite image, we can actually see the same species, Emiliania huxleyi, at work. And in this case, she's forming a huge bloom that covers multiple hundreds of square kilometers. And why do the satellites pick up the signal of such blooms? Well, that's because the free-floating coccoliths that are produced and are shed during such coccolithophore blooms act like little mirrors. So they reflect the sunlight at the surface of the ocean, and therefore the satellite imaging sensors pick up on those differences in light reflection. So these milky blue and green swirls are basically the effect of these coccoliths scattering the sunlight. So coccolithophores have two jobs to do. One is being part of the biological pump, and that's through the process of photosynthesis. They lock in inorganic carbon, CO2, into organic matter. And coccolithophores are not alone. There's many other phytoplankton groups such as diatoms, dinoflagellates, and cyanobacteria who do a similar job of pumping CO2 into the marine realm by producing the organic matter that mostly forms the base of the marine food web. So other organisms use that organic matter produced by the phytoplankton as fuel to operate their lifestyles, and actually through respiration then the CO2 is readily released again. So we're pumping it in, organic matter here, and pushing it out again through respiration on the other end. However, that chain is not so efficient, so also a lot of that organic matter travels to the deep sea, where it's also again respired by bacteria or other organisms that live in the deeper realms of the oceans. And only a fraction of that organic matter gets trapped in the sediments and is actually effectively removed for many millions of years from the system. And so the pumping effect is understood as that atmospheric CO2 gets effectively brought to the deeper levels of the oceans, where the respiration through bacteria releases CO2 as well, but that CO2 is no longer in direct contact with the surface ocean and atmosphere interface. So let's say it's building up in the deep sea, but it's not in direct contact with the atmosphere any longer. And so we're effectively pumping CO2 from the atmosphere into the deeper ocean, and eventually also a tiny fraction of that gets buried in the geological arc. So the strength of this pumping is a function of how many phytoplankton there are, so their abundance. But on a cellular level, if we think of a single cell as a little factory of photosynthesis, and how much a single cell may package in terms of organic matter, also the cell size matters in this case. And then abundance and cell size makes also a nice functional combination to think of how many generations or how much biomass can be built up over time. So coccolithophores have another very important job to do, and that is biocalcification. And this is often called the calcite counterpump. So we have a pump, the biological pump, and here we have a calcite counterpump. Why is it called a counterpump? Well, with biocalcification, we fix alkalinity, bicarbonate ions, into a solid, calcite, a mineral. And there, effectively, we are actually building in some carbon into a solid mineral that is, of course, no longer part of the soluble ions in the ocean. But at the same time, we also release CO2. So for every 2 moles of bicarbonate or alkalinity that is used in biocalcification, we have 1 mole of carbon being locked in efficiently in the solids, but another mole is released in the form of CO2. And that becomes this counterpump to the biological. When coccoliths rain down, like let's say marine snow, or through the guts of grazers, these coccoliths travel down quite efficiently also to the deep sea, where part of them are dissolved under the influence of the CO2 that resides in the depths. But also we get an accumulation of many of these coccoliths, and think of the chalks, so that we have hard evidence that these organisms are rock-forming algae. It's also good to note that actually the calcite burial far exceeds the amount of organic carbon that is buried in the deep sea. So, as we have the 2 pumps, one countering the other, it's very important to figure out what is the relative balance between the 2 processes. And that's often dubbed the pick-to-POC ratio, so the ratio between the production of inorganic carbon, i.e. calcite, versus the production of organic carbon, organic matter. And we can look at that pick-to-POC ratio also on a cellular level. So we can investigate modern-day coccolithophores, such as Emiliania huxleyi, which here is also shown in its single-layer cocosphere. So here you can see how tiny it gets. But here we also introduced a new species to our story, and that is Helicosphaera carteri. And you can maybe readily appreciate that Helicosphaera has much larger cell volume than Emiliania, and also has much larger coccoliths and very differently shaped coccoliths. And so on a cellular level, we can ask the question, how much pick-to-POC is produced in each taxon? And why we want to, let's say, spread over multiple species, or to investigate not only our poster child Emiliania, is because we know that the ancestry of Emiliania in the clade Isocoracidalis and the family of the Norellarabdacea, that clade split off already in the early Jurassic from all other coccolithophores that we know to live today, and that includes the Helicosphaera lineage. So basically, if you think of the family tree of life, and when different clades diverge and then have their individual evolutionary path, think of chimpanzees and the human species. The chimpanzees and human species split off only maybe 6 to 8 million years ago. So 195 million years ago is quite a long, let's say, long time to also expect that maybe these different lineages have different modes of adaptation and modes of evolving over this long time. So this is of interest to our story today. I've shown you these beautiful cocospheres taken by SCM, so intact cells, but often when we go to the fossil record, we only find individual platelets, coccoliths. Here, some reticulofenestrates shown. And so we barely or rarely find these intact cocospheres being fossilized. However, when we have the patients, and once you see them, you can't unsee them, so you can hunt for them and actually establish also in the fossil record the range of cell sizes and how that relates to the individual coccolith lengths that you find on these cells. So we have actually a very nice, fairly linear way of just measuring individual coccoliths and determining the ancient cell size of these species we measure. And this relationship holds in the modern day taxa, and in here we show the ancestry of Emiliania huxleyi, and in this case Harry McClelland and others used several strains of Emiliania and also Gipharocapsa oceanica, who are all part of the same family, to show that indeed the same relationship exists between coccolith size and coccolith cells diameter for these modern day strains, and that it's actually overlapping exactly with the fossil relationship that we established here. What they do add to the story is that in this case they had live algae growing in the lab, so they were able to actually measure the cellular pick to POC ratio, which is on this vertical axis here. And that they then were able to relate to the coccoliths that were formed by these organisms in culture. And they express it in the so-called coccolith aspect ratio here. And the aspect ratio is actually a very simple ratio between the thickness, in terms of calcite thickness, and the size of the coccolith. And here I'd like to talk about a pancake story, where if you make a lot of batter, think of that as your calcite batter, and we're baking American pancakes or French pancakes. So an American pancake tends to be quite small in its diameter, but thick. And if you were that French guy making crepes, you make very large area, but very thin pancakes. And so basically, the American pancake would have a high aspect ratio, and if scaled to cell size, would also have high pick to POC ratios. Whereas if we were baking crepes and very thin pancakes around a much bigger, let's say, beach ball, we would have also lower pick to POC ratios in that matter. So a lively example to imagine what we're doing here. But the good news is that, again, based on single coccoliths, we can have an estimate, an indirect estimate of the pick to POC ratio of those ancient algae when they were alive in the past. So how do we weigh a coccolith, or how do we determine the thickness of the coccoliths? Well, we applied Luc Beaufort's method that you can read all about in his publication here. And for Helicosphaera here, I will illustrate what that method entails. And the short of it is that here we see 4 individual fossil coccoliths of Helicosphaera morphospecies. And where it is very bright, the coccolith is very thick, so there's thick calcite. And where it's less bright, it will be thinner. Although in the case of Helicosphaera, we also deal with 2 types of orientations of the crystal elements in the coccolith, where the white ones in this case are the radial elements, and the gray, the vertical elements, so the VR model that Jeremy Young explains nicely in his talk. And that means that actually the gray that we see in these individuals is actually an underestimation of thickness. It's not really thin calcite, but we just basically can't measure it completely. So we have let's say, an index of changes in thickness, but we also have a very good way of measuring the area, because the entire coccolith is visible in the light microscope. So here I'll show you some of the results based on culture experiments where we add the Helicosphaera carteri. We took two strains and to add to the database that was supplied by McClelland and others. So we have the Emiliania huxleyi strains here, and then in the middle we have Gipharocapsa oceanica. So let's have a look at these 4 panels. The top panel shows lith volume, so the amount of batter or the amount of calcite contained in the individual liths produced in culture, versus either cell volume or this aspect ratio. And you can see that it's plotted on the ln, so natural logarithmic scale, so that we can actually see that relative changes on the smallest and on the largest scale are comparable, because it's like comparing mice and elephants, and let's say the relative changes in the size of a mouse would be, let's say, not visible on the elephant scale, and otherwise very confusing on the elephant scale. So you see that it's quite a nice and tight correlation, where small cells have smaller lith volume and also larger cells have larger lith volume. And again, that's also expressed in this aspect ratio in panel B. So if we look at the lower panel, we have again cell volume and aspect ratio on the horizontal axis, but here we're looking at the peak to POC ratio. And the short of it here, again, we see a fairly, let's say, fairly clear scaling of small things versus larger things. But I would like to stress that the spread of the peak to POC ratio here is much larger. And again, it's on the log scale, so this is quite a huge jump in peak to POC ratios. But that actually cell volume is very conservative. Let's say within a single strain, for each of these single strains, the cell volume is rather constant, constrained, but the peak to POC volume is much more plastic, so is much more variable. However, what we do see is that the larger taxa are always in this blue band, which would be a heavy calcifier, where the peak to POC ratio is above 1, meaning that basically the CO2 output through calcification exceeds the CO2 lock-in through photosynthesis. So I'll continue telling you a story of 15 million year history of these two lineages, and I will use the measurements of individual coccoliths to tell this story. We will visit two deep sea sites, one in the temperate South Atlantic and one in the tropical Indian Ocean. The RCC numbers here are the Roscoff culture collection numbers of the Helicosphaera carteri strains that we used as a benchmark for this story. So let's have a look at a time series. So we go from 0 today back to about 15 million years. And I show you two lineages, the lineage of the Helicosphaera and the Norellahaptasea, so the ancestry of Emiliania huxleyi. And they're on, let's say, the same scale, linear scale in this case. And each sample that you see, each dot, is actually an average of a minimum amount of measurements of 50. In total, we measured over 4,000 individual coccoliths for the Helicosphaerids and more than 300 for the culture collection or the cultured specimens. So a lot of measuring is involved. And when we then look at the overall patterns, in both lineages, what you can appreciate, I hope, is that over time, there's a selection for smaller cells. Here I notice also that for the Norellahaptasea, we have multiple sites, and the red data were collected by Clara Bolton and others, and the blue is the data collected for the temperate South Atlantic site. Same samples at the Helicosphaera in the blue curve. So the question is, why would this selection for smaller cells have occurred over time? And it's very, tempting and probably wise to look at patterns of paleoclimate evolution. So how did climate evolve as these algae were evolving or selecting for smaller cells over time? So here I give you an example of how sea surface temperature changed in the temperate regions in blue and in the tropical region in red. And you can see that the cooling is specifically pronounced in the temperate or higher latitude regions, but also that the contrast between the tropics and the temperate regions is increasing over time. And that has implications for how the ocean is mixing and how nutrients are made available also in the surface ocean for these phytoplankton groups. So there's several hypotheses linking, let's say, paleoclimate change to the selection for smaller cells. But they basically all come with the main idea that the selection for smaller cells is due to some type of resource limitation. So some type of building block becomes lacking or limiting for these algae, so that smaller cells who need less, have less requirements for nutrients, also on a cellular level produce less quota of organic matter, of calcite. So in a way, it's more efficient to be small under stressful circumstances. And so the hypotheses include some levels of, let's say, nutrient limitation or direct carbon limitation, because with cooling climate also atmospheric CO2 levels declined, and that may have imposed some limiting effects on photosynthesis itself. But it could also be an effect indirectly through cooling and lower CO2, that the weathering on the continents was much less efficient or strong, which leads to less building blocks and alkalinity nutrients and alkalinity being brought to the oceans as a sort of bulk inventory. So there's multiple research groups puzzling about these questions, and I think in recent years we've come up with quite a nice set of data to try and ground truth this story. So the other part of our investigation was how about the plasticity in the degree of calcification, or how did the biogeochemical output of these algae change or evolve over time? And that's where we went to, let's say, investigate this aspect ratio. And so here in colors, we see Helicosphaera again, the red from the tropical Indian Ocean and the blue from the South Atlantic. And you see they overlap pretty much and pretty, let's say, stable over time. So it seems that the aspect ratio in the Helicosphaera lineage is very conservative, not changing much. Whereas if we look at the similar data for the Norellahaptasea here in gray, you see that they actually changed from rather heavily calcified to much lighter calcified forms by the late Miocene. So much more plasticity or variation in the Norellahaptasea than in Helicosphaera. And this falls nicely in the narrative or the understanding of Helicosphaera being an obligate calcifier. So Helicosphaera, like many other coccolithophore lineages such as Calcidiscus and Coccolitus, they're heavy calcifiers, but they also need to calcify. So without calcifying, they don't survive. They can't, successfully reproduce and maintain large populations. And that means that with this conservative aspect ratio, they also had probably very conservative biogeochemical output on the cellular level. So the peak to POC ratio didn't change much. Whereas the Norellahaptasea have much higher plasticity in the degree of calcification. And this is also there's also evidence for that, because we have naked, non-calcifying E. huxleyi strains that do perfectly fine in all their, photosynthesis and division jobs otherwise. So there's a big difference between these clades. However, I've also shown you in the same pictures that Helicosphaera was a constant presence and constant contributor to the ecology over the past 15 million years. So it must have done something right. And the question is, what other adaptive traits could we propose for this lineage to, let's say, cope with resource limitation over time. So I'll close with this final slide and showing you Helicosphaera carteri. We know it all now. And showing you another cell next to it. And for non-experts, it might be puzzling why I'm showing you yet another shape and form. But this smaller cell with, let's say, tiny coccoliths on top of it is actually the haploid representative of the same species. So we're looking at Helicosphaera carteri either in its diploid life cycle, where we have 2 copies of the chromosomes inside the cell and the heterococcolith production, and then through meiosis, the life cycle can shift to the haploid stage, where we have a single set of chromosomes in this cell, and then through syngamy, these cells can again come back to the diploid life stage of species. And what we know about the haploid stage is that it often occurs in very oligotrophic conditions. For instance, in the Eastern Mediterranean and Adriatic Sea, you find the haploid stage of many coccolithophores only in summer, or more often, more abundantly in summer when it's very oligotrophic in the Mediterranean. And then in winter, when there's more storm mixing, the diploid stages are more present. So this might be a very efficient way for these obligate calcifiers to sustain their populations over time and only, let's say, pop up when there's enough resources to maintain diploid populations. So with that, I would extend some special thanks to the Phytoscale team members, most especially to my co-author, Luca Shupraha, who is the photographer of these magnificent images, and then the International Ocean Discovery Program, IODP, for supplying us with a wealth of deep sea records to study and study the evolution of life, but also the evolution of planet's climate over time. And last but not least, the INA and the very efficient Aurora science communication team for this invitation. And if you are interested in reading up on all the details, story that I was just telling you, you have free access to the publication on biogeosciences.net. So with that, I thank you. I hope to see you live somewhere else someday. And if you have any questions, don't hesitate to contact me through email.